Hello everyone, I am Alok Tongaonkar, one of the hosts for today's meeting. Welcome to today's talk on lessons and opportunities in smart large-scale networks and health applications by Professor Chen-Nee, Chuah. Before we start the talk, I would like to introduce the organizers of the talk, the San Francisco Bay Area chapter of ACM. This chapter was founded in 1957 with an aim to promote knowledge of modern computing, create community, support networking, and hiring. The annual membership is $20. Please consider joining by visiting sfbayacm.org. We organize 2 monthly meetings. The first is the General Computing Talk on the 3rd Wednesday of the month. The second is Data Science SIG on the 4th Monday of the month. We also have joint meetings with other ACM groups, as well as with AI Camp and ValleyML.ai. Here are the upcoming events. First is the ML Data Engineering Conference being held today and tomorrow. This features over 30 speakers from leading companies. The details are on the meetup page. Tomorrow, we have an interesting talk on Intro to Computer Vision, building object detection models and datasets by Sage Elliott, who's a machine learning engineer and advocate at SIGGIL. This is a joint event with Silicon Valley ACM SIGGRAPH. Next month on May 19th, we have another interesting talk on the COVID-19 conundrum, critical insights regarding cloud security posture of enterprises by Matthew Chiodi, Chief Security Officer of Prisma Cloud at Palo Alto Networks. Some logistics for today's meetings. Use Q&A in Zoom for questions to the speaker. Use chat on Zoom or YouTube for questions to the panelists. And announcements for job or other events can be posted in comment sections of the meetup page. It is my pleasure to introduce our speaker today, Professor Chen-Nee Chuah, who's the Child Family Professor in Engineering at the Department of Electrical and Computer Engineering at UC Davis. She has MS and PhD in Electrical Engineering and Computer Sciences from the University of California, Berkeley. Her past research interests include internet measurements, network management, and cybersecurity. Her more recent projects focus on applying data science and intelligent learning techniques to tackle predictive and decision control problems in societal scale networked systems, such as massive online social platforms, intelligent transportation systems, and smart health applications. She has led funded projects from industry as well as NIH, NSF, DoD, including multi-institution grants. She was a recipient of the NSF Career Award and was named a Chancellor's Fellow of UC Davis in 2008 and UC Davis Advanced Scholar in 2020. She's a fellow of the IEEE and an ACM Distinguished Scientist. So without further ado, I would like to hand it over to Professor Chen-Nee Chuah.
Thank you. Share screen. All right, can everyone see my screen okay?
Yes, we can.
All right. So thank you, Alok, for the introduction. Today, I would like to talk a little bit about the lessons learned and opportunities from the research projects that have been going on in my group for the past 20 years, working in the domain of experimental data science. In particular, as we apply that to large-scale networks problem, as well as more recently in the smart health domain. So first, just to give you a little bit of a background of my research interests. So I started out in 2002 at UC Davis, and even back then, I mean, my interest is in monitoring and measuring complex network systems. So I spent about 9 months visiting Sprint Advanced Technology Lab at the time and collaborate with them, the IP research group, to look at collecting data from production networks so that we know what's going on and we can do analysis on the routing stability, right? Both at a control plane as well as at the data plane, trying to understand the characteristics of wide area internet traffic. Now, at the time, I was actually looking at more from the infrastructure perspective, right? Because coming from a networking background. Now, as internet scaled, we start to run a lot of large-scale network applications as well. So around 2007, when Facebook opened up the development platform, one of my students actually started to look into developing Facebook application, and we realized that application developer at the server itself is also a measurement point. It gave us a window into looking at user interaction on online social platforms. So we started to look at network, but more at the logical level, looking at connectivity graphs, traffic distribution graphs, as well as looking at the how information cascade through the online social network platforms. Now, at the same time, as a side project, I also have collaboration with faculty in civil engineering and computer science, looking at intelligent transportation systems as yet another network systems, where we have network nodes are the intersections, and then we have the incoming, the lanes are basically incoming and outgoing queues in the system. Now, more recently, my group kind of moved to more the smart health domain, where we leverage IoTs and wearables to collect information about patients, and both inside the hospitals and sometimes also outside, and try to apply machine learning and AI techniques in order to do predictive analytics, disease diagnosis and prognosis, and also looking at the operation of the hospitals, right? So for example, I mean, this is yet another type of communication network we can look at, is how the different providers cooperate with one another by reviewing the files, enter the clinician notes, as well as the labs results for a particular patient, and cooperate to provide complex care to the patients, especially in cancer care, for example, for complex disease management. So across our projects, I would say that we are interested in innovations across the entire data pipeline, from layer one, where we acquire the data, till the actionable items. So when I say layer one, data acquisition, the first question we ask is, how do we collect data? Sometimes the data is not available yet. We have to instrument and look at measurement instrumentation in order to be able to collect those data to measure and observe events of interest. Now, in this context, sometimes the data is too large, right? There are too much amount of data, and then we have to come up with algorithms like streaming and sampling algorithms to help us handle the load. And sometimes there are cases where we're interested in events that you cannot measure directly. So we'll have to look at partial view or end-to-end view, and then use some inference techniques to infer internal characteristics of the system. And then moreover, in a dynamic environment, sometimes you also have to make the measurement algorithms itself adapt to new requirements, adapt to new operating conditions. Once we get the data, we basically deploy techniques from different domains depending on the problems at hand. If it is a time series signals, we'll borrow techniques from signal processing. We use techniques from machine learning and deep learning as well, in order to translate the raw data that we are collecting into insights and information and knowledge that we can use, which will help us in the third plane, which is the data-driven control. Now, from the context of the networks, internet networks, or the cyber infrastructure, a lot of the problems we are interested in are more in the resource management or resource provisioning space, or how do we improve the quality of service or quality experience for user by performing data-driven control. We also care about fault resilient routing or security detection. Now, when we apply this pipeline to other domain and other application domains, such as, say, intelligent transportation system, the third component, the data-driven control there, the goals might be different, right? So for transportation, it could be increasing throughput at traffic intersections, or you want to protect the driver, so we want to improve driver safety, as well as you want to minimize fuel consumption. Whereas for smart health, you have yet another different goal, right? The goal might be you want to do screening for a certain disease so that you can introduce intervention early. So as I mentioned before, our group look at these types of innovations across data pipeline in different application domain. And today I just want to talk a little bit about lessons that I learned over the years, applying this to the computer networks domain, and how that insights helped me approach this new domain, smart health, in the last 4 years. All right, so now let's look at the first part of my talk, right? Look at lessons learned from internet measurements work. All right, so internet measurements community, right? It's started really, the early research started in late 90s and early 2000s. At the time we call it internet measurements, we didn't, but really if you can think about it, it's really just big data over computer networks, right? We just did not use the word big data back then, 20 years ago. So at the time, the challenge is data access, right? It's hard to get data from production network because you don't own those network. So there was some initial research that was carried out at tier 1 ISPs like AT&T and Sprint that started looking to how to instrument their network so that they can collect control information such as the routing messages between the routers, as well as data traffic that is going on the access link and the call links, and trying to understand exactly how the network is functioning. And over the years, right, so back in 2001, there was the first internet measurement workshop where this kind of work started to get published, and there are presentations about internet measurement work. And slowly this community has grown over the years. By 2003, there is a first ACM International Measurement Conference held in Miami, Florida, and then it became a very mature, vibrant research community. Right now we have annual single track international conference on internet measurements like IMC and PAM, passive active measurements. And there are repositories, right, public repositories of data and tools, both active measurements and passive measurements that researchers and at large have access to. So for example, Write, CADA, Crawdad, these are just some example repositories that have data about the routing plane, data about traffic, and so on and so forth. So I would say over the years, the impact is this community has really enabled reproducible research. By sharing this data and tools, now other researchers can actually repeat the experiments and get the same results. And over the years, there is also some established ethical guidelines of how are we supposed to collect this data, right? How do we protect user privacy with the proper anonymization steps? How do we collect data in such a way that we are introducing the minimal disturbance into the system that we are observing, and so on and so forth. And I also see a lot of tech transfer between the research work into the industry. In particular, right, if you look at the talks in NANOC, North American Network Operators Group, and you can look at technology transfer between academia into network operators and equipment vendors and cloud providers and so on and so forth. So the first lesson that I learned, right, by working in this field of internet measurements, I would say in the first 10 years of my career at UC Davis, is that there is room for innovation in data acquisition itself. Even though our end goal is usually, I want to understand, for example, right, routing instability problem. I want to study, how does the traffic patterns look like, right, for traffic matrix estimation so that I can do traffic engineering. But the first part of the problem is to get the data. And how we measure data, right, that design of that instrumentation, that process needs to be cognizant of the application goals and operating constraints, because monitoring resources are limited, or you cannot possibly just measure everything under the sun. So this is actually a resource allocation problem. And any intelligent learning techniques can be applied to this phase, to the data acquisition phase as well. So just to give you context, right? Let's say we start with network monitoring and detection problem. Why is it so difficult? Well, the first problem or challenge is scalability issues, right? We have a lot of data going on in the network. It's already a lot to measure back then in 20 years ago, and now the problem is just getting worse, right? This is based on the 2021 report. We are going to have billions of devices, generate 236 exabytes per month of data, and that's going to overwhelm processor, memory, network storage, and also administrator, right, for operating this network. So scalability is a big challenge. Second, a lot of times, the events of interest that we want to measure could be stealthy, right? So for example, for security detection purposes, we are interested in actually MICE flows that generate very little packets. For example, for port scanning type traffic. Now, if you just simply use sampling approach to sample packets coming into your network to deal with scalability issues, most likely you're going to miss this MICE flows completely. And if you miss them completely, you can't even do any analysis, right? And let alone doing detection. Now, the other challenge is that the traffic, the workload keeps on changing. And our network operating condition also keep on changing. So the questions of where, how, and what to measure. For example, if you want to deploy the deep packet inspection box, where should you place it, right? Maybe at the beginning, when you're asking this question, you're interested in, let's say, the red flow. But then maybe later on, things have changed and you also want to measure the green flow. And so that is the almost like, having a moving target that you have to adapt your monitoring framework and monitoring solution to. So one of the key innovations from our group that I'm still proud of today is this concept called programmable and adaptive measurement. And this was back in 2007, we reported this idea in a SICOM paper where we said, well, maybe we should come up with a more flexible measurement plane where we can dynamically configure what traffic subpopulation we want to measure based on the application goals. But at the time, in order to do this, right, we have routers and switches are still pretty, I would say, it's hard to innovate because we are tied to what NetFlow can do and what are the software that is supported by a particular vendor. So in order to implement our idea or experiment with our idea, we need new software, hardware, and abstraction language. But luckily, right, this is quickly followed by the software-defined networking paradigm and a lot of work that went into developing OpenFlow. And it turns out that our solution is a perfect match for that paradigm. So we have a subsequent demo applications using OpenFlow. And the idea is that you could have a controller, you can design and change your SDN-capable routers into programmable network monitor. And you can design rules, measurement rules, right? That is part of your flow entries. So you can push all these TM entries into the different monitors to collect data, collect the statistics for a certain application. So one example application could be traffic matrix estimation, which is important for a lot of resource provisioning work. And based on the quality of your measurements and your estimation, you can update the design rules, right? Your monitoring rules and reinstall new rules. So this is just an example that I want to showcase because I want to stress that innovation can happen at the data acquisition phase of your data pipeline. And intelligent learning algorithms is also important here, right? How do you design and update these measurement rules? You need some kind of learning task. And in fact, that continuous learning is important. So back into 2016, when we tried to basically build a demonstration of our programmable measurements idea on SDN, we use traffic estimation as an example problem. And in order to estimate the full traffic matrix, we are allocating some TCAM entries in the routers to track some important flows, right? To measure some flows directly so that we can then fold that into the traffic matrix estimation problem to estimate the rest of the entries that are not measured directly. So we look at a single switch case, and let's say you have K is the number of measuring entries that you have at a switch. This is your monitoring resources that you can allocate. Then the question is which K flows, right? Out of all the N flows that you see at a switch, should you monitor? If you just randomly select it, which is the black dotted line, right? Not surprisingly, you are not helping much, right? Your average estimation error is still pretty high. If you do a one shot, meaning you're just looking at previous historical measurements, you can do multiple rounds, exploit, just to observe what kind of flows you have, and you try to rank them based on the largest to the shortest flow, and you optimize them and do it only once, you reduce the estimation error, but then you are going to miss out as time goes by because your traffic changes. And these key important flows that are important in one time instant might not be important later on, right? Maybe half an hour later or even minutes later. So the dotted red line is basically if we formulate this as a multi-arm bandit problem of trying to optimize and look at basically trying to have online learning algorithm, right? Can we do this better, right? And the answer is yes. If you have a continuous learning algorithms, right, both the red and the green line, you can actually get a better estimation error in solving the traffic matrix estimation problem. All right, so now let's move on to the second lesson that I learned. The second lesson is the importance of the data pre-processing part of things and featurization of your data and feature selection. Right, so the first question after you collect your data is how do you represent your data? What kind of features should you extract from your signals? What features are important and meaningful for learning? Now, and this is where I stress that it's important to leverage domain expertise depending on the target applications that you want the learning tasks are designed for. Right? And there is a terminology for this also these days, and then some people call it science-guided machine learning. And it's not only important to make sure that this is not only important to make sure that your target application will have good classification or prediction performance results. It also helps with interpretability because you know what you're looking for. So again, just to give you some case study and examples. So let's say we are collecting packet header from an IP backbone or wireless cellular network or just enterprise network, right? So your input data is the same. It's just packet header, right? So you can say you track the first 20 or the first 40 bytes of the packets. If your monitoring application is capacity planning or traffic engineering, then you most likely care about volume count. So with the same packet trace, you can look at the number of bytes per second sent, number of packets per second sent, or number of flows per second sent. And you are going to end up with time series that look like the plot here. And then you can basically apply different types of time series analysis or abrupt change analysis. If let's say you want to detect, sudden spike of volume for DOS attack, you could. I mean, you could look at wavelet transform of the signal in order to detect anomalies. Now, if let's say now your goal is different, you're interested in fingerprint applications, then from the same set of packet data, you won't be looking at just packet count or the byte count. You'll be looking at the source destination IP address and port number, and so that you can construct something like the traffic distribution graphs, right? Which nodes are communicating with which other nodes on a certain known ports. And based on this connection graph or the traffic distribution graph, you can potentially guess or estimate or predict what kind of applications are running there, right? So if it's BitTorrent, you have a mesh-like topology, and if it is email, you have servers, or web, you have a server base, so you're going to have more like a star topology. Now, third application scenario is, again, you have packet header, but maybe now you want to look at statistical packet or flow levels features, right? So you could look at inter-packet arrival times. You can actually look at the different packets and match that to a particular flow first, and look at flow level features. For example, how many total bytes or total packets a certain flow is sending. And if you are looking deeper into, let's say, the TCP level, or you can extract URL from the packet, you can also look at average URL length and other information. Now, why is that useful? Well, one of the applications could be to detect malicious wireless traffic. And this is some of the work that we did in collaboration with a wireless cellular provider. We want to look at, how much malicious traffic is there. Can we actually predict or fingerprint this malicious traffic only based on network traffic? So we look at a bunch of features, like what is the uplink and downlink tonnage, right? Amount of bytes sent. We look at the entropy of the destination IP address and proportion of failed connections, and things that you can actually these are features that you can extract from same type of packet trace that I talked about before, where you can build the graph from, or you can look at the volume count. Now, in this case, if we look at these statistical features at the flow level, we can actually differentiate the potentially malicious host and a benign host. Right? So this is for a totally different application. So as you can see, in these 3 different applications, we are extracting different types of features from the same type of data. They are all just packet headers. Now, the last lesson I want to share is, how do we choose model? Sometimes the choice of models are governed by the problem goals as well as practical constraints. So this is back in 2012 when we are interested in looking at user activity graphs on Facebook, on Facebook applications, gifting applications. And Facebook is an online social platforms, right? So naturally, there are a lot of graph models that exist for friendship graphs. So when we look at, what is the degree distribution for the user activity graph, right? Which in this case is directional, because A could send something to B more frequently than B sent it to A. We realize that the existing or the classical friendship graph actually fail. Why? Because the friendship graph assumed that link is symmetrical, right? You are friends. So A is friend with B, B is friend with A. That's it. So if we use that, it turns out that we can actually model the in-degree distribution of the activity graphs pretty well, right? That's the top graph. The degree distribution matches pretty well. But when we are trying to look at the out-degree distribution, there is a huge gap. Why is that? Well, it turns out that it actually costs something to send an invitation to a friend. You can receive from as many friends as you can, right, on an online social network-based gifting application, but there is a limit of how much you can send, because the online social platform like Facebook actually limit the sending rate, right? They don't want any users to spam other users. So it costs something to send, right? So as a result, you actually have to modify the existing preferential attachment model to capture that aspect of it. You have to place constraints on the outgoing invitation sent. And after that, right, we did some simulation to look at, how well does our modified model work compared to the empirical data. And it turns out that we could actually now then match the in-degree and the out-degree distributions pretty well. Okay, so that kind of is just a kind of intro to what I want to talk about in the second part of my talk, is that all these 3 lessons learned, right? from the previous projects helped me approach the more recent projects in my group on this in the smart health domain. In particular, like I'm going to basically look at 3 case studies, AI ML driven clinical decision support systems, where we look at time series data and patient data from in a hospital environment, especially for critical patient care, like ICU environment. And then I'm going to also talk about 2 other case studies where the input data now is image in the second case for the brain tissue phenotyping. And in the third case is the video as a data source. So this is a critical care, as one of my collaborators, Dr. Jason Adams from UCD Medical Center was telling me, right? It's a data-rich environment, but information poor. As the doctor walked in, this is what they are faced with, right? There are a lot of monitors, a lot of screens, a lot of data. And the ICU doctor has to quickly digest all of this in order to make a decision of how to adjust or how to prescribe a treatment for this patient. It could be changing a different setting on a mechanical ventilator machine. It could be changing the amount of fluid that you want to administer to the patients, as well as other drugs and lab work that need to be ordered. So moving into the future, right? The future of smart health, really, I feel like there is now a convergence of all these different technologies together. On one hand, with IoTs and wearables, you can continuously gather data, right? So there are some time series data that you can keep logging and for analysis. You also have electronic health record data, right? The labs, image, X-ray, MRI, and so on and so forth about the patient and patient history. Now, these days, right, with video visits and videos you can take in the clinical setting as well, you also have video and mobile data. And lastly, all this data, right, if you do apply big data analytics on this, you can do patient disease diagnostic screening or prognosis. And in the future, we can also couple this with robotics and AI and have automatic control with human in the loop of some of the support system, right? Especially the clinical decision support systems. It could be AI assisted in the sense that we are not completely try to automate it, but it provides enough intelligence to help the physician or the nurse and the providers to introduce intervention in a timely manner. So for example, for the ventilation decision support system, it could actually tell the nurses or the respiratory experts whether or not it's time to adjust the setting on the machine for a particular patient. And then same thing for fluid administration, you could also imagine having a continuous monitoring and fluid control device that will help the patient, that will adjust according to the patient condition without having someone being on the bedside 24/7. So there are a lot of opportunities in smart health applications. So the examples I give you is more from the critical care perspective in ICU, pediatric or the newborn ICU, right? So PQ and NICU, so that you can perform early detection of critical events. You can do patient prognosis, and you can help the physicians or the care team decide what to do next, right? In the emergency room, you could also think of having this kind of an analytics going on in the background to decide how to route patients. So this is a resource management issues for a hospital. Now, there are patients where, these are chronic disease management that involve not just things that are happening during the checkup, but also outside of the outside of the hospital, at home, and in daily lives. Like, for example, cancer patients have a lot of complex issues involved with them. One example is blood clot development. And so you could use all the patient data in order to, and the medications taken and history to predict risk for blood clot development. Diabetes is another example, right? For glucose monitoring, now it's very easy to get time series data, continuous monitoring. Now, the other side of thing is the lab, on the lab, right? So we have pathology and radiology. I mean, they have a lot of research and development going on there as well. One example that I want to talk about here is in the Alzheimer Disease Center at UC Davis, where the researchers are looking at brain tissue slides and trying to phenotype, right, the how disease like Alzheimer progresses, right? And they're trying to look at the image and look at the certain protein that are hallmarks of Alzheimer's disease, and where are they located? Is it in the gray matter or in the white matter? So these are just yet another venue where having, if we have those data and we can apply machine learning and AI techniques, it could help phenotype the neuropathology researchers are really keen on adopting the analytic data pipelines. And then there's also lab work that are involving drug administration, drug effects, and so on and so forth. And then lastly, there is behavior monitoring, right? So for lifestyle tracking, so tracking of cognitive decline and behavioral health like autism or depression. So there are a lot of opportunities out there. So back to this, connecting to the first part of my talk, I mean, there's definitely innovations that we can make in both the applied and fundamental data science when we try to apply that to smart health. So in the first plane, in the data collection plane, right, I talk about IoTs, but there is also interesting questions regarding system engineering, regarding, if there's a lot of data that need to be collected, how do we decide, right? How much data is enough or how much data to keep? And if you have high frequency data, you also can decide, I mean, if we need all the fine granularity or can you afford to downsample so that, you have less data storage. Sorry, let me move back. And then in terms of data science, the second layer, once you collect the data, I mean, that's the same thing, depending on what we're interested in. There are time series analysis or machine learning and deep learning techniques that we can leverage there. And then lastly is the translational AI and learning-based control, right? And most likely we'll still have human in the loop because it's important to have the domain experts to decide and make the final call in the patient disease diagnostic and prognosis process. So we had different sources of data, right? In the hospital or clinical environment, you have EHR data, you could have continuous physiological signals that are collected by sensors and wearables. You could have image and videos as well. And then there are new opportunities here, right? There are some data that you might want to be able to monitor, but they don't exist today, and you just need some smart engineers to design new wearables and sensors for that scenario. So I just want to kind of share with you some of the ongoing work in my lab right now in the area of AI-driven critical care. So the first project is a DoD-funded project looking at ML-driven ventilation management as well as acute respiratory distress syndrome, ARDS detection. So ARDS is basically a wet lung condition. It affects 10% of the ICU admission. 25% of the mechanical ventilator patients develop ARDS. And once they have ARDS, yes?
Oh, sorry. There was a question that came in a few minutes ago. Was asking, how do you know traffic is malicious? I don't think they're talking about the medical cases, but on some of the earlier network analysis. So talking about how do you know traffic is malicious for malicious network traffic.
Got it. Okay, so do you want me to answer questions now or wait until the end for the Q&A?
Do you have a preference one way or the other?
I don't. I just, depending on.
Go ahead and answer questions. If we get too many questions,
Okay. Yeah, I'm happy to answer that. So I know I did not give you a lot of details in that part because my focus is more kind of showcasing, for the same type of data, you can use it to answer different questions and by extracting different features. So in that problem, our ground truth is based on security logs from deep packet inspection boxes and IDS that the cellular provider have, right? And so those, we look at the intersection of all the reports from their different IDS boxes that it installed and their different DPS, deep packet inspection box. So those are what we call the ground truth, right? That's the best ground truth we can have. Now, our goal was to try to see if, without looking into the packet content, because DPI is deep packet inspection, right? You look deep into the packet. So our goal is looking at just the network traffic itself, just look at packet headers. And we are trying to see if we can find enough how should I say, enough features that are of interest, that have predictive power, so that we can fingerprint malicious versus benign hosts, just purely based on network traffic. So that was the goal of the exercise. So how do I know it's malicious? It's because our group was collaborating with a cellular provider, so we have access to the security logs. So I hope that answered that question.
Thank you.
Okay. So, all right. So now this project is, in terms of ARDS, we know that it's a critical condition. And if the patient actually develop ARDS, depending on whether it's mild, moderate, or serious ARDS, unfortunately, it also translates to a high mortality rate, right? It's 35% to 45%. So it was important to detect these conditions early so that the ICU doctor can then prescribe intervention. Okay. So the second project is also funded by DoD, is looking at adaptive therapy for resuscitation after trauma. Right? So this is a case where in order to stabilize the patients, we need to administer fluid to stabilize their blood pressure. And we want to be able to predict the fluid response. And then the third project is funded by NIH, looking at, this is actually looking at babies in the NICU. So we want to develop an ML-driven screening for detecting heart defects in infants before they go home. All right, so I will start with the first project, right, as an example. So this is ML-driven ventilator management. So the very, very first problem that we look at is very simple, right? So if we can monitor the mechanical ventilator waveform, we know that the setting has to be set right. Otherwise, the patient might be fighting the ventilator for air, right? So this is what we call the patient ventilator asynchrony problem. So mechanical ventilation is life-saving, but if you don't set the setting right, it could be harmful or even fatal to the patients, because the ICU patients are already very sick to start out with. So PVA, right, is basically referring to the case where the patient demand is not met by the ventilator assistance. And here are just some waveforms to give you some examples of what I mean by the demand does not match the ventilator, right? So pressure curve on top is what the mechanical ventilator pressure pressure level is. And then the one below is the airflow, right? Inspiration and expiration for the patient as a result of the help from the mechanical ventilator. So when you see things like double trigger, it's just meaning that the mechanical ventilator thinks that the job is done, but the patient actually wanted more air, and it triggered another, right? Another inspiration and expiration, and that's what we call double trigger. There are cases also where you have delay termination, right? The patient is done breathing. I mean, it's going to the exhale phase, but the pressure is still continuous. And sometimes they are failed trigger, right? So these are just example of patient ventilator asynchrony that could be harmful to the patients. So in order to understand the rest of the talk about what features make sense, if I have access to mechanical ventilator waveform, let's look at the normal ventilator-assisted breath. So under a normal condition, right, this is like a gold standard. You have the pressure curve on top. This is from the mechanical ventilator, and the patient is getting this kind of airflow, right? So you have inspiration and expiration. And from here, you can, these are the parameters that are of interest to the ICU doctor, right? What is the peak inspiratory pressure, PIP, and what is the positive end expiratory pressure, and what is the tidal volume for inspiration and expiration part of the breath. All right, so in order to actually address this problem, we actually have to build things from scratch, right? So right now, the mechanical ventilator does not output any waveform. So at the beginning of the project, the first step is we actually had to retrofit it and use a Raspberry Pi to just send the waveforms, raw waveforms, to a server so that we can store it and we can do some analysis later on. And then, once we have the waveforms, we can downsample it, we can sample it, or we can just use the 50 hertz data points that we have for the mechanical ventilator waveform to do different kind of analysis. And then the third part is we also have a student kind of developing an iOS app for the doctor so that they can actually see some of these waveforms in real time. Right, so whatever analysis and the output of analytics can be displayed to the physician so that they can actually give us feedback, right? So they can mark and say, yes, your detection of the abnormal breath is correct or incorrect.
A question?
Yes.
On the Raspberry Pi, what logic are you executing on that? I mean, is there some kind of fast Fourier transforms or digital signal processing on that, or is it just?
So at this moment, no, we do not do any processing per se. We are just collecting the data from the mechanical ventilator and use the Raspberry Pi to stream it through wireless to the wireless network so that we can stream it to the server. But in future, this is the edge device that potentially can do some local processing. Right now, everything is at a server.
Okay, yeah. So this is happening more in the server for all the analytics then. Okay.
For now, yes. But that is actually our future goal, is to have some of the edge device doing the inference.
Sure.
All right, so the aim one, right? So we talk about different ways of different types of situation where your patient ventilator asynchrony can happen. But to start out with, right, we try the simpler problem. We basically look at what is the most common PVA that can happen, right? And by talking to the physician, we were told that, okay, double trigger is quite common, breath stacking is quite common. Double trigger is when, you trigger two breaths instead of, because the pressure from the mechanical ventilator machine stopped like too early. The patient actually wanted to inhale more air and then trigger the second phase. And then the breath stacking is where the volume, the tidal volume for inspiration and expiration don't match up, right? So for this analytical pipeline, so as mentioned before, right, part of it is data acquisition. We spend quite a bit of time just to figure out how to get the data. And then, why do we, well, first of all, why do we even think about mechanical ventilator waveform as a data source, right? Because typically for ARDS patient, one way to determine that the patient has ARDS is actually look at chest x-ray, right? So if the patient is getting sicker and sicker and they suspect that the patient may have ARDS, they will order a chest x-ray. And then based on the report of the chest x-ray, then the physician can decide whether or not this patient has ARDS and whether or not they want to introduce intervention. Now that obviously has a delay, right? So it's not happening in the first hour of admission of the patient. Mechanical ventilator waveform, on the other hand, these are continuous waveform. It's already there, right? The minute the patient is put under mechanical ventilator machine, we can start collect this data. So the advantage of this approach is it's ubiquitous, it's continuous, and it's objective, right? It's just waveform, it's a time series data. That's why we go that route. And so now we can do waveform analysis and algorithms. We can do feature extraction. So that's the second lesson learned from before, right? We need proper featurization. So in order to work on this project, it takes a team of multidisciplinary folks. And my students from my lab and my postdoc actually work closely with the ICU doctors and nurse and respiratory experts in order to understand this. We are trying to understand exactly how should we featurize this signal. So we look at features that are what we call expert features, right? So they tell us what is important to them. We look at total volume, inhale, exhale, for example, inhalation period and exhalation period. These are just the simpler ones to understand. And there are other metrics that or features that we need to extract from the waveform as well. And then based on this, right, after we extract the features, then the rest is a little bit more vanilla, right? It's more kind of standard. We can fold it into a machine learning classification problem. We can use off-the-shelf machine learning models or deep learning models. And our goal here is to look at, for example, at a per breath basis, we can see which of the breath is normal and which is off target. Or we can also look at the condition of the patient over a longer period of time. We can also get over an hour across multiple breaths and decide whether or not this patient has ARDS. And if the patient has ARDS, potentially we can also keep on monitoring over a period of day or even weeks to see how the ARDS prognosis processes, right? Does it get better? Does it get worse? So at the beginning, we only have a pretty small data set. We have about 20 patients, 350 breath, and we have 2 clinicians manually annotate this breath to tell us whether or not they are normal, they are double trigger, breath stack, or sometimes there are artifacts as well, because if the patient turn and the patient cough, there is suction, that can result in anomalous breath as well. So in terms of the statistics that we have in our data set, we have 6,000 normal, 500 normal breath, and then we have a handful, right, 752 of DTA, and about 2,000 of the BSA, right? And then in terms of feature selection, we have clinically relevant statistics that we derive from the raw ventilator data. There are 64 base features. And then we perform feature engineering. And we also look at, what happened if we just throw all these features in and then we do feature engineering, and it turns out that we can select 7 optimal for BSA and 11 optimal for double trigger. And then the next experiment we did was, if we don't do this statistical analysis and sensitivity analysis and feature engineering, we just go by what the doctor said, right? So we take the 3 most telling sign, the 3 features, what will be the results? And then we look at off-the-shelf models, k-mean clustering, multi-layer perceptron, gradient boost, random forest, extremely random tree classifier, and also an ensemble of these multiple models. And this is a multi-class classification problem, right? We analyze data for both DTA and BSA at the same time, and then we add features chosen by the experts to those chosen by statistical methods. Now, we also have a class imbalance issue here because we have a lot more normal breath than DTA or BSA, especially for DTA is very minority, is a minority class. So we apply SMOT to do upsampling of the minority class. In terms of performance metrics, we're interested in true positive rate or sensitivity, specificity is a true negative rate. Sometimes, for this type of smart health application domain, we're also interested in precision or accuracy. So, these are the typical metrics that we'll look at. for smart health predictive analytics problem. And here are the detection results for the different models that we experiment with. And it turns out that, some models are better for detecting the breast stack anomaly. The other one are better for DTA. So we look at an ensemble, which gave us a pretty good AUC in general, right? A trade-off between sensitivity and specificity. So if you're interested in more details, these are in our 2018 paper in the methods of, and I have the full reference in my last slide. All right, so that is just looking at a per breath basis. So now I just want to talk a little bit about, how do we kind of looking at across breath and use a mechanical ventilator data, waveform data, to detect ARDS, which is actually happens to be also relevant in this pandemic. So for ARDS detection, we have 50 ARDS, 50 non-ARDS patient data. And the way we, called out we have more data than this, but in the end, we call out this set because we want to make sure we have at least 1 hour of the VEN data in the first 24 hours of either the patient being put on mechanical ventilator for a normal patient, for the normal class, or for the ARDS patient, we want to have that 1 hour of VEN data in the first 24 hours after the ARDS diagnosis. And then we extract 8 expert features per breath based on conversation with the ICU doctor. And then for each breath, we can extract these features. But if we are doing ARDS detection, right, is a condition, we want longer observation period, we actually look at the average physiologic feature for each read, and a read is 100 breath. And we actually do sensitivity analysis of how many breath in a read makes sense. And then for each of these read, we can basically classify whether or not this patient has ARDS or not. Now, because this is a dynamic condition, right? So one of the ways for us to basically predict that final output label is to look at this written different reads. We had classification per read, and then we do a majority voting, right? So for a particular patient, you could have, different amount of VEN data, and for each 100 breath, we have a label that our model predicted. And then after the entire observation period, we look at what percentage of those labels actually says this patient have ARDS. If it is more than 50%, then we classify as ARDS patients. Otherwise, we say no, it's other category. So this is reported in our recent paper in critical medicine, and we try different off-the-shelf models, and we get pretty good sensitivity and specificity results with pretty good confidence interval as well. Now, because our data size is pretty small, right? So we have 50 ARDS, 50 non-ARDS. For random forest, we actually also look at different ways of testing. We look at whole out tests of 70-30 split. We look at bootstrap, and the results actually hold. And we also did other sensitivity analysis by reducing the data set further. And of all these models, eventually we chose random forest to focus more on because it's pretty robust to outliers, and it also does not tend to overfit. So I saw that there was a chat. Was there a question? Sorry, I missed it now.
No, I don't see any questions right now.
Okay, all right, thank you.
Oh, somebody asked how large of a random forest was used?
We have about 500 trees.
Okay. And was model description an issue to try and describe the reasons of a model? There could be global model description or local to a specific forecast.
Say that again, sorry. Can you repeat a question?
Was model description something of interest, either describing how the model makes its forecasts or classifications overall, or on a record by record basis, like if you give a model a particular record and it scores or inferences on that record to give local reasons for that particular analysis.
So interpretability is very important for the physician. In this case, actually, we look at expert chosen features. We also have statistical feature. We also do statistical feature selection. And then we also compare with, because like, for example, random forest model, right, also compute the Gini importance. And so we try to compare like whether or not those that are deemed important by the model match with what the physician think are important. And the answer is yes. I mean, a lot of these cases, experiments that we run, once we report the Gini importance and look at the, rank the features importance based on that, and we go back to check with the physician, actually match with what they intuitively think are important features for predicting the condition.
So the ranked features would give an overall model description.
Yes.
And then for a given forecast, then is there a way of identifying for this record, this set of data that you're analyzing, particular features that are important for that record?
Correct.
Okay. Another question came in from YouTube, was asking the introduction of analyzed information into the ICU environment. Is UX also part of the research solution? It seems information needs to be presented in a very critical way, like in avionics.
So yeah, so again, this is just this is not translational yet. I mean, we are really looking at very preliminary study, right, of a small data set that we get from an ICU setting. We are not actually doing this ML-based screening yet, right? Because this is not done in practice in the ICU. This is pure research at this stage, and we finally have some initial results and we publish it. But our next step is actually we have to test this against data sets that are collected in other settings, right? Hopefully other hospital settings, so that we can test whether or not our model, our trained model, can actually work with heterogeneous data, input data that our model hasn't seen before. And then after that, we might actually look at potentially having a shadow ARDS screening happening, because since we already have the app developed for the physician for them to view the analytics, we can then look at some of this quality check of whether or not our screening, our model for screening ARDS is effective. And then there will be multiple other steps involved before we can even test it in a clinical environment. So yeah, we are still quite a ways off, I would say, to that final goal of testing it in a clinical environment.
Okay. And another question from YouTube, William was asking, do you input patient-specific parameters as well, like body weight or body mass index?
No. So in this, it's purely just one data source. It's just a time series data from the mechanical ventilator. Now, future version of the models, obviously, we can fold in other information we have about the patient. But right now, for this work, we are only looking at time series data. We want to know, we want to push the envelope, basically. We want to know how far can we go by just looking at a continuous time series data from the mechanical ventilator machine.
I see. And this is a question from me. At some point in the future, do you think you might use inpatient, in-office genetic data and add that to your time series so you have a better context of the patient?
So I'm not well, in future, if the genetic data is available, but also that question, I think is better. I have better go back to ask my collaborator, all right, Jason, and see if it makes sense clinically. Because I'm not a medical expert, so I cannot, tell you what information about a patient is most relevant here in this case.
Thank you. I think you're caught up on questions.
Okay. All right. So now our next step is, well, so far we were looking at expert generated features. These are features that experts tell us are important that we can extract from the time series data. We are also curious, right? Well, what if we just apply a black box, right? We have the time series data, we throw it into deep learning. Well, what will happen? So we try this as well. So we look at the best sequence, treat it as, because it's a time series data, we use 1D CNN. So we look at a sequence length of 224 data points. And the reason we chose that is when we do a characterization, 224 data points capture roughly about a breath, right? So of course, each breath, the length is different, even for the same patient. And then we actually try CNN. We also try LSTM and CNN plus LSTM. And we get pretty good AUC results as well in terms of the area under the curve as a trade-off between sensitivity and specificity. For CNN and LSTM, it doesn't do as well in terms of specificity, and it takes longer to train, and it tend to overfit. But a 1D CNN works pretty well. So now we are curious, is that we want to know what the deep learning is learning really, right? Because here we don't do any feature extraction or feature engineering. So we try to look at GradCam, we try to look at the frequency component of the signal that we have, and for the different class, right? What is deemed important from our CNN model? And it turns out that for the non-ARDS patient, the normal patient without a condition, a lot of these decisions are based on low frequency signal. And then for the ARDS patient, the more important component is a high frequency signal. Now, and then we go back and have this conversation with my collaborator, Jason, and he thinks that it's reasonable in the sense that what low frequency signal means is the envelope, right? So you have this envelope, the envelope of a breath, basically. And high frequency signal is because of all this transient, right? High frequency component of the or the fluctuation of the flow or the pressure curve that we see as an input. So that's what we notice from our deep learning model exploration. And then we actually also look at feasibility study, right? Just now you're asking, whether or not the Raspberry Pi is doing more processing, right, than just collecting waveform and sending it off. Now, so we have a model. We want to look at lightweight edge device that can run inference model. So we actually try to implement our model on NVIDIA Jetson Nano, and it does some raw waveform pre-processing, pre-trained model that we install there. And then we have a new instance of Breat coming in and try to infer it. And we found out that we can do, we can actually spit out the answer in sub-seconds, right? We can have single breath data coming in, and then we can decide whether or not the patient has ARDS using our trained model and have the results ready in 0.13 seconds. And one breath is roughly a minute of ventilator data. So this is usable. Okay. All right, so any questions before I move on? I saw there is a chat, but I wasn't sure.
The chat just had somebody posting a link to a paper.
Okay, all right. All right, so now the other two projects, I also want to talk about the second project because so far the ARDS detection problem, we're looking at one time series data, and the features are pretty kind of well defined, right? The physician tells us what they are, and then we can just apply normal, signal processing techniques to really read out those values and extract those values. Now the second project for EPAC is a little bit messier, because the input data here is actually from animal experiments. And these are really sick animals, right? These are pig that have either hemorrhage or sepsis. And yes.
For the audience of data scientists who don't have a medical background, when you say fluid response, could you elaborate a little bit?
Say that again. Oh, what is fluid response? Okay.
Yes.
So this is when the patient is really sick, or in this case, the pig are really sick, and you need to administer fluid. And fluid response just means that after you administer the fluid, you actually result in improvement of the blood pressure.
So when you say fluid, this is like an IV?
Yeah,
like
IV, yeah. Yeah. So in these experiments, we are collecting data. And when I say these experiments are done in Travis Air Force as well as University of Utah by the other PIs on this project. And then we collect the data like temperature, respiratory rates, and blood pressure waveforms and ventilator data. So obviously, we also need to do data pre-processing and feature detection here. So for their signals, we downsample it to 100 or 200 hertz. We need to remove major noise by smoothing or low pass filter. And then more importantly, for the features that are of interest to the physician, we are interested in things like the systolic peak, diastolic peak, and the dichrotic notch in the blood pressure pulses. So these are the whole pipeline that is involved for this project. Right? There are many steps involved. The actual ML model development and validation, right, is on the right-hand side. And to be honest, that is a small part of our effort. I felt like for the entire project so far, the bulk of our work is in the first part, the data acquisition, pre-processing, visualization, and to extract the features of interest. And why is it so messy? Well, this is an example. So we have the arterial blood pressure and the central venous pressure waveform from the pig. And then this on the bottom right, this is basically our reference signal, right? So every 60 seconds, they experiment by administering 100 ml or 100 cc of the fluid to the pig. And then they back off, and then another 100, and so on and so forth. So every of these 5 microboluses together is called a macrobolus. There's one event, there's one observation. So the question is, if I observe these waveforms that happen, ABP and CBP, during this period, right? Or maybe just up until before I administer the fluid, versus now I'm trying to observe it after I administer it, do I see an improvement in the cardiac output, in the blood pressure, right? So there's a cardiac output and there's a stroke volume. So for this analysis, right, we went back to the doctor and basically we need to clarify, right? What do you mean by being fluid responsive? And our answer is that, based on medical journal and their expertise, right, they will view it as an improvement if the cardiac output improved by 15%. And so that's what we go by, right, to generate our ground truth, right? So we have a positive response if the blood pressure improved by 15% before, when we compare before and after the fluid administration. Otherwise, we say that it's a negative class. All right, so now let's look at the data, the signal that we have. So we have this ABP and CVP signal, right? These are blood pressure pulses. And we're interested in the systolic and diastolic, right? So the red dot and the blue dot peak. And then we're interested in dichrotic notch. Now, it turns out that the dichrotic notch detection is really challenging. And then, of course, we are also interested in, with various other features that we can extract once we detect these 3 points. We might be interested in area under the curve up until the dichrotic notch, and then between the dichrotic notch and the dystolic peak, and so on and so forth. So, these are the waveforms that we are dealing with, right? So obviously the first step is remove noise, because there could be a lot of randomness in between as well. And there might be different local minima as well, right? So when we want to detect the dichrotic notches, right, it makes sense to smooth out the unwanted notches. So in order to do this, right, so the first step is for the ABP waveform, we know that any signal component that is greater than some accepted threshold can be considered noise. Okay, so we can throw that out. We can have some low pass filter to do that. But we have to be careful in selecting the filter because we want to preserve sharp edges, right? So that, and I apologize for the spelling mistake here. I mean edges, right? EDGES here. Because we want to be able to call out which points are the dichrotic notch. So we ended up using a Savinsky-Golay filters because it's a moving average filter. It smooths things, but it preserves sharp edges. All right, so why is it so difficult? Because the ABP waveforms in real life are messy, especially the ones from sick animals, right? For example, sometimes there is no local minima at all, right? It's hard to figure out where your dichrotic notch is. And sometimes they are multiple. And then sometimes they are fake dichrotic notch that, you know, your algorithm might pick up. And sometimes after you run the smoothing, you can't even figure out whether or not there is a dichrotic notch. So because of this, we actually have to look at rule-based algorithm to encode domain expertise in this so that we ensure that only the actual dichrotic notches are detected, right? So the first step is we try to find the distinct peaks, and even the peaks, sometimes you have inflection points and false peaks. So you have to make sure that we find the right peak. And after you find the peak, you find the valley, right? You find the local minima between the two peaks. And then after that, you limit the search area between your S, BP, and DNP to find your dichrotic notch. And the location of dichrotic notch, it really depends on the timing of the aortic valve closure, right? So we have to end up calculating first and second derivative of the ABP waveform and then perform some kind of final light set filters on all these candidates to find the actual DN. And these are, you know, typically, if you look at the medical literature, I mean, there are different classes of dichrotic notch. And that's why it's so hard to actually to code up an algorithm that can automatically extract these features for us. So in order to do that, all right, and I want to just back up a little bit, we ended up having to code out this web interface called extract so that we can display this signals from different animals. And then we have physician to actually go in and mark some of these dichrotic notches and also the peaks for us so that we can use this as a ground truth to test our algorithm, our feature extraction algorithm. So after we have that resolved, right? So now we have a set of features. We can go on and do the feature engineering, model development, and validation part of it, right? So we do, we select features with predicted powers. We do correlation filtering, so filter out features that may be highly correlated. And then because we have a small data set, we actually do model selection using nested cross validation. So in the end, we have about 4 sepsis and 14 hemorrhage model animals. We have we look at the different macro bolus events as one observation. So we have about 224 of them, and only a small percentage, 26%, are fluid responsive. Those are the positive class, and then we have 74%. Majority of them are non-responsive. And so we do have a little bit of class imbalance there. So we can so we ended up experiment with, whether or not we need to do minority upsampling or majority downsampling to keep the data set balance. And then in terms of features from the 2 waveform, we have 52 initial input features. And after we do the statistical feature selection, we basically reduce it down to a total of 9 features in the end for our final model training and testing. And then we look at different models, logistic regression, random forest, and gradient boosted trees. And in our case, random forest offer us the best performance. And in terms of the correlation filter, the threshold of 0.7 also resulted in a sweet spot for us. And so we can get an ROC AUC about 0.89 in this case. And then this is for the left-hand side. I mean, these are the ROC curve in terms of trade-off between the sensitivity and specificity. And then the right-hand side is the precision and recall trade-off. And then similarly to the previous case, we are also curious about, what happened if we throw this into a deep learning model. So we use an existing off-the-shelf model from this paper, right, that look at LSTM fully convolutional network. And we have two types of time series data. So again, we basically look at batches of 224 data points, and it gives us roughly compatible ROC AUC compared to the RF, the random forest. So this is actually the current state of our project, but it's still ongoing. And we want to further improve the prediction and accuracy by incorporating ventilator waveform and other metadata like the respiratory rate, temperature, and other things that we have as well. Okay, so that kind of, conclude the part of the talk that look at, mostly time series data and using time series data to develop the pipeline, data analytic pipeline for disease screening purposes. So now I want to switch gear a little bit to talk about another two projects in my group that also that look at image and video instead as input. So this project is in collaboration with Dr. Brittany Duggar from the Alzheimer Disease Center at UC Davis. The ADC Center actually, they has, one of the most diverse collection of human brains for Alzheimer's disease and related disorders. So in terms of the, for the neuropathologist, they are interested in looking at amyloid beta plaques and other type of proteins that have different density distributions in gray matter and white matter. And so the current approach is to separate the gray matter and white matter by manual annotation. So Dr. Duggar and her team will manually annotate it on this digitized tissue images using software like Aperio, ImageScope, or QPath. And then they recently, have applied CNN to detect those plaques automatically. And so with the manual annotation of this different region, now they can count, right, how many of these plaques are in the gray region versus the white region. So it's one step towards phenotyping human brains. But it's labor intensive. Any annotation, right, manual annotation is labor intensive. So our question was, can we automate this step with image processing or deep learning pipelines? So we ended up developing an end-to-end pipeline for Dr. Duggar to automatically separate the gray and white matter. Right, so it involves some image processing in both the pre and the post-processing part of it. And then in the middle, we actually ended up using CNN. So this is the deep learning-based GM, gray matter, white matter segmentation pipeline. First of all, these are very, very high resolution image slide. So in order to scale it, so that it's scalable, we actually turn the problem of segmentation into patch-based classification. So we look at small tiles at a time, and then for each small tile, it's just a classification problem. We want to classify it to either is background, tissue, is gray matter, or is white matter. And then after we have done that, we turn it back to a pixel-wise segmentation.
Was it a semi-supervised learning approach where you had some people do initial labeling, but not exhaustively on lots? And then
yeah, you're jumping ahead. So we did do that too. So it's coming. So at the beginning, we just try to do supervised machine learning first. So we have the label data. We have about 20 slides label. And we try to, actually in total, we have 40 now. So we compare the different models, right? FCN, UNet are well-known models for medical image segmentation problem. And then we also look at, ResNet, and then my student also try to introduce another layer called NCRF, neural condition random field, so that you can incorporate spatial relationships between the neighboring patches to improve the performance. So we have some preliminary results, right? In terms of, this is an image segmentation problem. So we look at IOU as our metrics. We just recently submitted a paper to IEEE Journal of Biomedical and Health Informatics. But as you can see on this, this is probably easier to digest in the table. is we look at the different region, right? So we have gray matter and white matter. We want to look at the precision score accuracy in F1. And actually, it turns out that for our problem, FCN doesn't work as well, right? So UNet works pretty well and very close to our pipeline of using ResNet patch-based approach or NCRF. But in terms of the training, in terms of stability, actually UNet, sometimes it doesn't converge. And so we ended up focusing on ResNet. Okay, so back to this question about semi-supervised learning. Yes, it is one of our challenges, right? So first of all, for supervised learning, our challenge is that different annotators, they could annotate the same slide differently. So there is inter-expert variability right here. Like for example, I show you two images here with two annotators, and the green line and the yellow line don't always match up, right? So even for supervised learning problem, there is this question of how do I encode this uncertainty in my label data set? And then the input data, as I mentioned before, these are high resolution image, right? So it's 60,000 by 50,000 pixels digital images. That's why we actually have to use a patch-based approach. We try to train on partition regions and further downsample it to run faster on a limited GPU memory. And even then, our image processing pipeline takes about 40 and 50 minutes to run per slide. And the training phase takes 10 hours, right? So there is still work to be done here, right? For us to actually optimize the pipeline further. There is large variance in the WSIs, right? So sometimes, because these are brain tissue and it depends on the cross section and how it was sliced. So it's in terms of heterogeneity, right? This is another challenge, right? How do we train our model with enough representation that it will actually work most of the time to properly segment the white matter and the gray matter? Limited label data set is a problem. And so we currently, actually, my student just submitted another paper to the biomedical analytics conference on using a combination of self-supervised and semi-supervised and self-supervised learning models to cope with this problem, right? So when you have limited data, but we also have a bunch of slides, they are just not annotated, can we actually leverage semi-supervised and self-supervised learning to perform this segmentation task? And the answer is yes, we actually got pretty good results. So hopefully, I'll be able to talk about it more in the near future. Okay, so that is kind of the current stage of this project that is, again, using machine learning and deep learning model, but looking at a slightly different type of data input. I mean, this time is image. And then in the last part of my talk, I want to talk about our NIH project that look at video data, right, for autism detection. Oops. I'm sorry. I think something happened here. Can you still see my slide?
Now we just see you, not the slides.
Okay. All right. So I think something happened. For some reason, my PowerPoint failed on me. So let me restart. Sorry about that.
Well, this way we know it's live in a real presentation, right?
I'm sorry?
I said this way we know it's a live, real presentation.
That's right. Live presentation always has some hiccups, right? Okay. Let me go to the right slide first. I think part of the problem is that all these images and slides, they are pretty huge. So sometimes PowerPoints are not happy. Oh, okay, let me. All right, I don't want to share it, but I want to go here. All right, hold on a second. Sorry about that. All right, let's see, here we go. Okay, let me share it again. Okay, does this work?
Yes, we see slides. Thank you.
Okay, good. All right, so this is the last part of my talk. This is an NIH project that is in collaboration with the Mind Institute at the UCD Health Campus, as well as University of Kentucky, computer vision expert there, Professor Samson Cheong. And the goal is to apply ML models to perform early detection of autistic spectrum disorder using video data. So for, I mean, the whole motivation for the work was that, I mean, Sally, right, Dr. Sally Ozanov is one of the renowned autism researcher at my institute, have been looking at the video data for, for her patients for a long time. And the motivation came from, in general, the median age of the first parental concern for autism is 18 months. And 30 to 50% report concerns before the first birthday. So in terms of the diagnostic age, typically, right, it happens between 18 to 36 months. So, but then studies have shown that if you have earlier detection, you can introduce earlier treatment and you will have improved outcomes. So her goal was to figure out a way to push that diagnostic age as early as possible. And she was wondering whether or not there is a way to use computer vision and machine learning techniques directly applied to the video data that she has so that we can do this detection earlier. So she actually has over 300,000 minutes of video. I mean, these are videos of children, right, both healthy and the ESD group that comes to the clinic and they have signed consent forms. And so we can analyze the data as researchers. And so in her lab, she had her team hand code, some of this ground truth. So basically, there are humans that watch these videos, recordings, and then try to look at events of interest. And in particular, right, they look at these 4 events, right? Look face is the event where whether or not the child's face is oriented toward the on-screen partner. Look object is if an object like a toy is introduced, whether or not the child's face is oriented towards the object. Smile, whether or not it displays some positive effect with any intensity. And then vocalization. And so the hand-coded ground truth is basically telling us like which minute of the video, right, this look face event or look object event start, and then when does it end. So for all these videos, we have this label data of basically duration and frequency of these events, right, for the videos that are associated with a particular child. So over the years, I mean, she has published many papers that look at, correlating these events with an autistic risk, right? So these 4 are basically behavior traits that could help differentiate the normal group, the control group, and the ESD group. But again, I mean, hand-coded ground truth means it's a labor-intensive procedure. So the goal of the project is whether or not we can have AI model learning with human in the loop. So now we start out with a labeled video data set, right? That's the red box that we have. We can train a model so that in future, when we have new uncoded videos, hopefully our model can automatically identify segments of the video that are most informative. And we can have then, right, a annotator to look at those and add that label back to our label video data set, right? So we will improve our data set, but eventually the goal is completely have our model, improved model, to be so confident that we can automatically just code those behavior based on the raw video. And with that coded behaviors, now we can actually build a classifier to predict whether or not it's an ESD or a control group. So machine learning and computer vision techniques have been used in ASD studies for a while. I mean, there are other, active research groups looking at this problem as well. So some use this computer vision and machine learning to study dyadic behaviors like joint tension, eyes gaze, measured by eyes tracker, repetitive movements. So there are research groups at Georgia Tech, UC San Diego, and UNC looking at this. There's also research groups that look at manually coded labels for autism prediction. So for example, right, at Stanford, there was a team that looked at videos that are uploaded by families, and then they crowdsource it, right? They actually have human raters answer a bunch of surveys that are related to, autistic behavior or non-autistic, and then they apply machine learning on those hand-coded survey to predict autism. So our pipeline is slightly different in the sense that we are trying to use a raw video and we try to look at behavior detection. We are trying to detect those 4 behavior that we talked about a few slides ago. Right now, we actually have 2 pipelines. One is looking at image-based deep learning, and the other one is the facial key point, right? Extracting facial features like, the eyes, the mouth, and so on and so forth. The current data set that we have, we look at a subset of the videos. We train it based on 100 children video. There are 403 video clips, depending on, these are taken of the same children over different visits. And then we have a validation and a testing set. So this is a relatively new project. We are in our year 2. So these are some of the initial preliminary results that we have. So one of the events that are of interest is look phase. So these are example results that from our pipeline for the look phase classification. So on the left-hand side is the actual kind of frame from the video looking at the expert labels that say, okay, this child is actually looking at the provider. And then on the right-hand side, right, so the blue are expert labels, because remember we have coded ground truth. We know the start and the end of the event. And then the red is what our model speaks, the label that our model produces, right, that predicted that this is actually a look phase event. Now you can see that there are cases where they lined up pretty nicely, but there are also cases where our model will pick up that it is look phase, but the expert label is not there. And it turns out that after we went and revisit this, there are cases where it is a false positive, but there are also cases where our model successfully picked up events, the look phase events that are missed by the expert, the annotator, the manual annotator. So these are pretty new results. We actually have a paper published in HealthCom reporting some of these initial results based on our image-based deep learning as well as the facial key point-based method to detect the different events. So far, we are doing pretty reasonable with the small look phase. We still have to improve on look object and vocalization. But these are ongoing work. So, I mean, hopefully, yeah, with more time and insights gained from our initial exploration, we can improve on the performance. So moving into the future, I mean, for this project in particular, right? So we are looking at automated detection of behavior. We hopefully this can replace the time-intensive hand coding. But then on the clinical side, we also hope that eventually we can move towards mobile apps for ASD screening. And hopefully this will help improving access to care, reduce the bottlenecks and disparities. All right, so I guess that's all I have prepared for today. So some concluding remarks. I think there are a lot of fun and impactful research to be done in the smart health domains. And as I mentioned before, the innovations can be across the data pipeline, right? Starting from instrumentation of introducing new sensors, new wearables, new edge device, to the signal and image processing step, the machine learning and deep learning development, definitely. And then in the end, how to put the AI-assisted clinical decision support system together with human in the loop. So I want to acknowledge a lot of my collaborators. They have been very patient with me and my students, answering our questions and also guiding us in understanding the physiological model. So there's Jason Adams in the critical care medicine at UC Davis, Austin Johnson in emergency medicine, and then a bunch of other pulmonary and critical care medicine doctors, Irene Brooks. And then Heather is the collaborator in the pediatric department for the project on detecting heart defects in children. And then Dr. Dagger from Alzheimer Disease Center, Sally Osner from Psychiatry and Mind Institute, Nicholas Anderson from Public Health in Davis, and then Samson Chung from University of Kentucky. And then some of these fund projects actually started with some seed funding from CITRUS and CSTC, and then later on, we have funding from DoD and NIH. And then these are just some of the papers that I reference for the work that I presented today. And with that, I'm happy to take questions.
William asked a while ago, I didn't want to interrupt, but are those features hand selected? I think they were talking about somewhere. This was at 8:29, so that was about 10 minutes ago.
So do you know which slide? Are we talking about these project or earlier project?
I think it was this child. Well, I'm not sure if it was, well, I guess, Tom, if you want to post, did you happen to remember what slide they were speaking to for the question? While they're doing that, just curious, for some of my vision recognition projects, I outsource to vendors to do labeling. So sometimes, I don't know if that would compete with the labor of grad students, but sometimes there's other options for that. And then like with Mechanical Turk, sometimes you have people who do reviews, types of things for more quality checks and controls. So I guess, had you considered any other places besides your people doing it, outsourcing labeling?
So this labeling part is already done, right? Because these are the database that we have from the Mind Institute.
Oh, okay. So for the faces, that was labeling.
Yeah.
Okay.
So for this data set, we already have the labels. It's just that we are used. That's why we can use supervised learning, because we have the hand-coded labels. So we are trying to evaluate our computer vision and machine learning pipeline against that.
Okay. So Anna asked a question for the ventilator project. What kind of interface is available at the ventilator? Like, was there a USB with the digital waveform data or something else?
So the mechanical ventilator waveforms, we actually have to hook up the Raspberry Pi at the back end and stream it out ourselves, because right now it doesn't output to anywhere. But I'm sure the vendors might be working on that, and they might export it somewhere later on. But it's just that at the moment when we started this project, the mechanical ventilator we have in the hospital, it did not export it, the waveform, anywhere, and it's not easily accessible. So we just have to basically stream it out ourselves.
Okay.
Yeah.
And then on YouTube, William had asked, he said the features was the, let's see, PASM linear project.
So
PASEMAN earlier project, I guess. I'm not quite sure. I think just earlier project.
So I would assume that is about the EPAC or the ARDS?
Probably the ARDS.
Okay.
So just was asking about the, let's see, were the features hand selected?
Yes. So the features that we talk about, when I refer to them as expert features, those, the bottom one, right? So the tidal volume, expiration and inspiration ratio, I mean, those are selected by the doctors. And then based on this, we also look at 64 base features that we can derive from the waveform, because it's a breath waveform. So we look at just the common ways of describing it, right? So we have expiration time, inspiration time, we have the area under the curve for the two sections. And then we also look at the difference of the current breath compared to the previous breath and things like that. So some are selected by the doctors, and then using that, we expand it and look at whatever features that we can extract from the waveform. And then we do the feature engineering.
Okay.
Yeah.
And let's see, Carl was asking for the autism video, was there audio for the vocalization, or was there a problem separating parent-child sounds?
There is audio for it as well. Yes.
Okay. And any problem separating the sounds of the parent or the child?
So right now, again, because the hand-coded labels are done by humans, right? So they do have that labels of the vocalization from the child. Okay. For our part, we will have to actually separate it ourselves and know which part is a vocalization by the child and which part is by the parent. So far, actually, to be honest, that's not an issue. Our problem is more of how to incorporate the temporal data, because I mean, by just looking at snapshot, right, we can look at facial, key point extraction and things like that. But it is the video, so over the frames, you do have temporal information as well. So we are actually currently looking into that.
So over time, are you using anything like a sort algorithm or different kinds of momentum of objects over time? Like sometimes in my vision projects, I'll have people walking across a room, and 2 out of 20 frames, we might drop, but then we know people don't teleport. And so as they're walking across the room, we do like a time series analysis. So using, a type of momentum analysis and overlap of images of frames, of successive frames. And we have an idea, oh, maybe we should go back and look there. So sometimes we can do some post-processing. For stationary objects, we know that the object tends not to teleport away if we're missing in a frame in our detection. So sometimes there's some analysis over time that helps improve accuracy. Was there anything like that you were doing with the faces?
So right now, we do not do that yet. What we do was, because we are interested in specific events, right? So like look face. So at all the frame, you can look at detecting the child's face, you know where the eyes are, right? You know where the eyes are looking at. And so even across that, you can roughly know how long across the different frames, how long the child is looking at the object. So that's the duration of that event.
Right. Yeah, I mean, that would be in a label.
Right.
In the analysis of that, but okay.
Yeah. I mean, right now the analysis, we are mostly focusing on the duration of the event and the frequency of the event. But moving into the future, what you are suggesting totally makes sense, because we want to incorporate that as well.
Okay.
Yeah.
Another question from Harsh. For the project using the NVIDIA Jetson computer vision, you have a stack of different devices or just one? Are you using open source computer vision tools or what CNNs?
So currently we are actually looking at just our the 1D CNN, and this is not a computer vision. This is just for the time series data, right? I mean, we are talking about the ARDS feasibility study. So the model is actually pretty simple, right? For this
one with the NVIDIA Jetson then? Okay, yeah, I see.
This is the one, yeah. So this is actually for just time series data. So we don't have to worry. Even this, actually, with the 2 GB memory, right? We had to worry about memory. I mean, how many breaths can we actually stream and store, and then we can run our model on it. And my student actually ended up having to use virtual memory, and we're still playing with it.
So what was, and this is my question, so what was executing on the NVIDIA and what was executing on the ARM?
Right now for this one, I believe the 1D CNN model is on the NVIDIA.
Okay, and the ARM is just a transport.
Yes.
Okay. Carl asks, for autism, it seems like the prediction was before human ground truth.
Can you explain that? What do you mean by prediction is before human ground truth?
You had the blue and red lines, and it seemed like the predictions were before the human ground truth on many.
Oh, I see what you mean. Okay, so that's the timeline you mean for
Yes.
the video segments. All right, let me go there so that I can look at it. All right. So this is actually across time, right? For the different different frames. So sometimes our prediction happened before the human labels, right? So this case, for example, and I hope everyone can see my mouse, right? You can see where I'm pointing at.
Yes.
Okay. So yes, so this is a case where actually our model picked up the event first, right, earlier than the human annotator. And then there are cases where the human annotator, like in this case, it picked up that label first before our model does, right? But most of the time, yes, our model seems to pick up the event earlier than a human annotator. So
Okay.
So is that a question about interpretation of this graph?
Well, I was just wondering if that was showing sometimes you can outperform human.
Yes, so we suspect that that's the case, and that's why I was saying just now that some of this misalignment, meaning like there is totally no human labels, we actually go back and look at those segments together with Dr. Ozanov, and we found out that in some of the cases, this is false positive, meaning this is a false positive on our model side. But there are instances where it is truly a look face event that the human annotator missed. So that's the encouraging part, right? The promising part is that our model actually managed to detect the events when a human annotator misses it.
I was just wondering also if maybe there's some built-in offset between what humans think versus what the computer thinks, and
Right.
you know, maybe humans are naturally decide later that it's occurring.
Yes, so I mean, this is, you know, it reminds me of the research we've done before with the security, malicious traffic detection part of things, right? There is really no real ground truth per se. This is the best ground truth we have right now to compare our models with, but then we realized that we will have to go back and revisit this because there could be human errors here, right, in the labeling process. And there is also delay, right? We are human after all. Sometimes we see something and then we try to annotate that this is the start of the event, but there could be some delay too, right? And so it's not 100% accurate ground truth in that sense.
Well, when you did this, were you playing a video and people would hit a button, and so that would add a delay automatically, and you didn't stop the video, they didn't home in on exactly when they started and end, or was it just kind of play and you just hit buttons as it was playing?
So as it is playing, right, the annotator will note the start of the event, and then it will mark the end of the event. So when it first look at the camera or look at the parent, and then when it stops looking at the parent, right, and they note the time point in the video, and then based on those time point, you can have the duration of that event, as well as like the frequency of a certain events in the video frame, in the video file.
But they're not jogging the video back to find exactly which frame they think it started.
No.
So that's a inherent delay.
Yes, there is inherent delay in it because they are not computer scientists and engineers doing the annotation, right? I mean, they just look at it and mark the start and end, but they don't look at frame level.
Thank you.
Okay. Are there any other questions? Anybody else wants to chat something or type something in the chat?
I did have another question.
Uh-huh.
For the ventilators, were you looking at all at a physical human lung modeling, you know, to perhaps supplement or the algorithms would then look for differences between what the model was versus what was recorded?
So very good question. So we haven't done that for the ventilator project yet, but we did do that for the EPAC project. So remember I talked about how hard it is to annotate, to actually extract the dichrotic notch from the waveforms. So we actually recently also have a side project looking at a model-driven approach, right? So just using the understanding of the underlying physiological model of how, like, you know, how a heart works, right? Our human hearts works, and to model it as a system and knowing that, right, if I were to generate a synthetic waveform of blood pressure, how would that look like? And what would be the peak? What would be the value? What would be the dichrotic notch? So using that model-driven approach, we also try to do that in order to figure out where the dichrotic notch is. And then we try to compare that with our rule-based approach. So that's definitely one direction that is of interest in our lab, is to look at a model-driven approach and see if it can help inform us in some of the things that we do, whether it's for feature extraction or in modeling it, the ML model development for classification. I see that there are activity in the chat.
Why might the random forest model turned out to be the best for the two studies?
So for the two studies, we have experimented with different models, right? So as you can see in our table, actually there are a few of those, right, gave very comparable performance. And the reason that we actually ended up, you know, looking at random forest specifically is because when we try to further stress test it, right, by purposely reducing the data set, there are some models that just fall apart, and then because they basically overfit. But random forest, we find is pretty resilient to that, and also resilient to outliers. So whether or not it is the best, I mean, I cannot say because we did not exhaustively search, right, for the best model per se. I mean, we are using off-the-shelf model to see, this is a feasibility test of will this work, whether or not just using one single source of data, like the time series data from mechanical ventilator machine, whether or not we can predict it, the ARDS condition. So, I mean, that's what we are looking at right now.
Okay. Carl, did you want to ask any other questions?
No, thank you.
Okay. No, that's good. Okay. If there's not further questions, then I'd like to thank you and invite the audience to give a speaker virtual applause. So thank you very much for speaking at the SF Bay ACM.
Thank you. Thank you, everybody, for your time and for your questions.
Okay. Thank you very much. Bye.
Thank you.